this video, we are going to learn about an experiment called estimation of protein by Biuret method. This is a quantitative test aimed to estimate the concentration of protein by Biuret method. Principle: the peptide bound nitrogen of protein forms a violet colored complex with cupric ion in alkaline medium. The intensity of the color produced is measured at 540 nanometer, which is proportional to the concentration of protein solution. Reagents: Biuret reagent, standard protein solution 5 mg per ml, bovine serum albumin or BSA in distilled water, test solution. Procedure: for working serial dilution, pipette out 2, 4, 6 and 8 ml at unknown quantity of standard BSA solution into a series of labeled test tubes. Make them up to 10 ml with distilled water in each test tube. Mix well and then test solutions were taken from each test tube. 4. Making test solution of concentration 0.8 mg per ml, 2 ml from 8 ml standard BSA plus 2 ml distilled water plus 2 ml Biuret reagent. For 0.6 mg per ml, 2 ml from 6.6 ml of standard BSA, 4 ml distilled water plus 2 ml Biuret reagent, like that. For 0.4 mg per ml, 2 ml from 4 ml standard BSA plus 6 ml distilled water plus 2 ml Biuret reagent. For 0.2 mg per ml, 2 ml from 2 ml of standard BSA plus 8 ml of distilled water plus 2 ml dilute Biuret reagent. Blank: 2 ml distilled water plus 2 ml Biuret reagent. Absorbance is measured in a colorimetric setup at 540 nm against blank. Plot a graph by taking the concentration on x-axis and absorbance on y-axis. Here we could see a graph. Estimation of protein by Biuret method. Scale x-axis given, y-axis is also given. We will get a straight line like this. Analysis of unknown sample. Take the sample in a cuvette and measure the absorbance at 540 nanometer against the distilled water or blank. Plot the absorbance on y-axis and draw a line parallel to the x-axis. Then the point where this line to x-axis give the concentration of the unknown sample. This can be calculated by the formula: concentration of sample is equal to absorbance of sample by absorbance of standard into concentration of standard. Once again, take the sample in a cuvette and measure the absorbance at 540 nanometer against distilled water and blank. Plot the absorbance on y-axis and draw a line parallel to the x-axis from the point where the line to x-axis give the concentration of the unknown sample. This can be calculated by the formula: concentration of sample is equal to absorbance of sample by absorbance of standard into concentration of standard. Result: concentration of unknown sample A from calculation and from graph, same value was got. Now some other aspects related to biostatistics. Different types of bar diagrams. Simple bar diagrams. A simple bar diagram is used to represent only one variable. Figure of scales, production, population, etc. for various years may be shown by means of single bar diagram. Since the bars are at same width and only the length varies, it becomes very popular in practice. Then subdivided bar diagrams. The bar is subdivided into different categories. Number of students in various courses in various colleges may be represented by a subdivided diagram. Then multiple bars. In a multiple bar diagram, 2 or more sets of interrelated data are represented. Then percentage bars, particularly useful in statistical work, which require the portrayal of relative changes in data. The length of the bars is kept equal to 100% and segments are cut into bars to represent the components percentage of an aggregate. Then deviation bars. Deviation bars are popularly used for representing net quantities of excess or a deficit, net profit, net loss, net exports or imports, etc. Such bars can have both positive and negative values. Positive values are shown above the line and negative values below it. Then broken bars. In certain series, there may be wide variations in values. Some values may be very small and others very large. In order to gain space for the smaller bars of the series, longer bars are broken. Two-dimensional diagrams. As distinguished from one-dimensional diagrams in which only the length of the bar is taken into account, the two-dimensional diagrams, the length as well as Width of the bar is also considered this. The area of the bars represent the given data. The important types of diagrams are rectangles, squares, and circles. Then pie diagrams. A pie diagram is a diagram in which data are converted into a scale of 360 units and a circle is subdivided into compartments band on this. A very common use of pie chart is to represent the division of sum of money into its components. For example, the entire circle of pie may represent a budget of a family for a month, and the sections may represent portions of budget allotted to different types of expenses. Then pictograms. There are picture used in representing statistical data. These are not abstract representations as lines or bars, but relate to picture count of data that are dealing with. Pictures are attractive and easy to comprehend, and such as this method is particularly useful in presenting statistics to laymen. The pictorial symbol is very carefully, it must be self-explanatory. Then cartogram. These are statistical maps used to give qualitative information on a geographical basis. They are thus represent spatial distribution. The quantities on the maps can be shown in many ways, such as through shades or columns, by dots, by placing pictograms in each graphical unit, and by placing the appropriate numerical figures in each geographical unit. Graphs of frequency distribution. The manner in which the frequencies are distributed over the different classes is called the frequency distribution of the character under study. A frequency distribution can be presented graphically in any of the following ways, like histogram. A histogram is a set of vertical bars whose areas are proportionate to the frequencies represented. While constructing a histogram, the variable is always taken on the x-axis and the frequencies on the y-axis. Each class is then represented by a distance on the scale that is proportional to the class interval. Then frequency polygon. A frequency polygon is a graph of frequency distribution. It is particularly effective in comparing two or more frequency distributions. There are two ways in which a frequency polygon can be constructed. One, by drawing a histogram. A frequency polygon is a graph of frequency distribution. It is particularly effective in comparing two or more frequency distributions. There are two ways in which a frequency polygon may be constructed. One, by drawing a histogram from the given data and then joining by straight lines and midpoints of the upper horizontal sides of the rectangle with the adjacent ones. The figure is obtained is called frequency polygon. Some statisticians, however, prefer to close both the ends of the polygon by extending them to the baseline. This extension is made an area under the polygon equal to the area under the corresponding histogram. Another method of constructing frequency polygon is to take midpoints of the various classes and then plot the frequency correspondingly to each point and to join all these points by straight lines. Frequency polygon has a special advantage over the histogram. The frequency polygons of several distributions can be plotted on the same axis, thereby making certain comparisons possible. Histograms are preferable when classes are few and frequency polygons when classes are numerous. Then smoothed frequency curve. A smoothed frequency curve can be drawn through the various points of a polygon. The curve is drawn freehand in such a manner that the area included under the curve is approximately the same as that of a polygon. The objective of drawing a smoothed frequency curve is to eliminate as far as possible all accidental variations that might be present in the data. Frequency curves usually show a single hump or mode value with the highest frequency and can be distinguished from one another by means of four characteristics of the central value, the spread of the curve around the central value, and the geometry or the symmetry or the departure from its termed skewness and the excess or deficiency of frequencies into the center and the two extremes compared with the flanks called kurtosis. When different components of data are to be shown, one can show them by means of sectors of a circle where the angles of sectors are proportional to the respective measurement of the different components. The data to be represented through the total of 360-degree measurement of a circle. The total frequencies or values are equated to 360-degree and then the angles corresponding to component parts are calculated. After determining these angles, the required sectors in the circle are shaded. Such a diagram is called a pie diagram.